Mhm. Okay, well, thanks everyone for joining us today for another QAAFI seminar. Let me just pull up my notes. So my name is Dr. Karen Massel, and I'm a research fellow in the Center for Crop Science in QAAFI, where I mostly study cereal crop improvement using new breeding technologies such as gene editing. Next slide, Brad. First and foremost, I'd like to do the acknowledgement of country. I acknowledge the traditional owners and their custodianships on the lands in which we meet today. I pay respect to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognize their valuable contributions to Australia and to global society. Next slide, Brad. Before we get started today, just a few housekeeping things. The seminar is scheduled from 12 to 1, so Brad should conclude around 12:45, and we'll have times for questions. Please put any of your questions or comments in the Q&A. We're not monitoring the chat function, so I won't see them if they don't go into the Q&A, and I will address those at the end of the seminar. So next slide, Brad. It's my pleasure to introduce today's speaker, Dr. Bradley Campbell. He's a research fellow in QAAFI. He's been with UQ for over 10 years, studying various things about plant molecular biology. His research involves the use of genomic tools for crop improvement with an emphasis on sustainable production of crops. He has extensive research experience focused on genomics and its relationship to diversity in both the agricultural and health settings, where he's got projects studying the aerobiome of different species and its relationship to allergies. His current projects, which he's going to discuss with us today, involve the genotyping of the Pacific Islands in vitro Taro collection for germplasm preservation and breeding, where he's investigating the infrared spectral cameras and their applicability to Taro salinity screening. So I'm going to hand it over to you, Brad.
Thanks for that introduction, Karen. And I'd just like to thank QAAFI for giving me this opportunity to present the work that our research group has been focused on at the moment. So to get the ball rolling, the title for the talk I'm going to give today is Evaluating Salinity Tolerance in Diverse Taro Wild Relatives to Enhance Food Security in the Pacific Islands. So just to give you a bit of background about what taro is, because not everyone may know, but taro is an important staple food crop, particularly in the Pacific Islands, but also in many countries around the world, such as Asia, South America. It's quite broadly used. There are 4 taro species in the Pacific Islands. Colocasia esculenta is what's true taro and is probably the most commonly eaten and is most widespread. For those who don't know, taro is a root crop like yam or potatoes. It's vegetatively propagated, and taros tend to differ in the types of colour of their corms and stalks, the stickiness of the corm, and the shape of their leaves. So a lot of the work we've been doing has been in collaboration with countries in the Pacific Islands. And we do a lot of collaborative work with an organisation called the Pacific Community, or SPC. And that's an international development organisation owned and governed by 26 countries and territory members. And they approached us originally to start collaborating on work focusing on one of their most important cultural staple crops, which is taro. So to give you an idea of the taro genome, it's about 2,405 megabases. Taro is probably one of the world's oldest crops. Evidence suggests it goes back as far as 28,000 years, back to the Solomon Islands. In terms of centres of domestication, it's still unresolved, but they believe it's either in northern India or New Guinea, where the areas of most diversity are found. Taro belongs to the family Colocasia and is a highly polymorphic species. Taro has a basic chromosome number of 14 and 2 cytotypes, diploid with 28 chromosomes, or triploid, which tend to be sterile, and 42 chromosomes. So you can see generally it takes up to 12 months for taro to reach full maturity before harvesting. And you can see in the picture here to my right, this is a picture of our resident PhD student, Gemma Restel. And just for context, not that Gemma has an inordinately large head, but these corms get quite large, and you can see how big they are in proportion there. So taro is grown globally, but it's still considered an orphan crop. You can see in here in the left-hand table that many countries around the world, from South America to Asia to even Africa, all produce taro. And in this case, they all have their own various field collections. But it's the Pacific Islands has the largest genetic conservation collection of taro in the world. And they have a large in vitro collection held at Fiji and Suva, consisting upwards now of up to 2,000 accessions. And as you can see here, they have various collections from all around Asia and the Pacific Islands. And this collection was started in the 1990s because a pathogen called taro leaf blight almost wiped out the industry, particularly in Samoa. And it was felt that there was a huge demand for genetic diversity in order to tackle such problems in the future. So a project called TaroGen was put together. And in fact, Ian Godwin and Emma Mace were key collaborators in that. And Emma was one of the first to do the first genotyping for the collection using microsatellites back in the 1990s. So in terms of the nutrient profile of taro, it's considered to be rich in nutrients that can provide important health benefits. Taro root has more than twice the fibre of potatoes. The carbohydrate content in taro root is what's called a resistant starch, which means it has beneficial aspects for things like reducing the risk of diabetes. There are high levels of potassium in the taro root, a mineral that helps to control high blood pressure by breaking down excess salt. Taro root is high in antioxidants, high levels of vitamin C, vitamin B6, vitamin E, and many other immune system boosting properties. The only downside to taro is it contains a bitter tasting compound called calcium oxalate, which are like crystalline hairs, that if you eat it raw can actually cause serious irritation in your throat. So it's always preferred that you cook taro before you eat it. So you can see here in the pictures to my right, taro, particularly in Asia, tends to be used in desserts or soups, but in a lot of Pacific countries, taro is roasted much like sweet potato or other yams or potatoes. So what is the economic value of taro? Well, as I mentioned before, it's an orphan crop. So you're looking at most a worldwide export value of 166 million US dollars. China is considered the top exporter. The United States is the top importer. And Nigeria is the biggest producer of taro in the world. In terms of the Australian context, taro has been grown in Australia for over 100 years, but there has only been a significant industry since the 1990s. And an annual Australian production is estimated about 1,000 to 1,500 tonnes. So it's a very small industry. And as you can see here in the map of distribution, taro is grown mainly around northern Queensland in the subtropics. And that's because basically it's a high rainfall tropical crop. So what are some of the problems that are facing the taro industry? Well, several reports indicate that there's significant levels of genetic erosion. Before taro leaf blight in the Solomon Islands occurred, taro and yam were the principal crops. But in Samoa in 1993, as I've already mentioned, the accidental introduction of taro leaf blight devastated production. Today, sweet potato accounts for most of the land devoted to food production, and evidence suggests that many cultivars are no longer grown. In New Guinea, taro production declined due to population pressure, plant diseases, and the introduction of high-yielding species. And in Micronesia, this decline was caused mostly by pests and diseases. Today, taro ranks behind breadfruit, yam, banana, and imported rice as staple food. Where taro is still cultivated, there is a trend towards growing a smaller number of varieties, selected or bred for high yield and monoculture, which is a problem because that's essentially what led to the taro leaf blight devastation in the first place, the constant use of monocultures. So that's an issue that the industry has to start taking a lot more seriously. So when we were approached by SPC originally to start working on taro, one of the big problems, well, many of the problems they faced, but one of the big aspects was they were worried about some of the effects of possible climate change that could face the industry. And in particular, they were worried about things like drought and salinity, but also other aspects like nutrition and that sort of thing like that. And I guess the question is, what does drought look like or salinity look like in the Pacific context? And that's something we have to think about. But there also are public health issues that are important. Increasingly, taro is being replaced by processed foods in people's diets, leading to increased incidence of diabetes. And there's also an issue of just the conservation of the species. Right now, Fiji is the only place that is maintaining significant collections of taro diversity. And it's hoped that in the future, that can become a permanent resource that can be used worldwide. There's also an extension and education aspect to the projects we take part in. And it's quite clear that the Pacific community has not quite kept pace with the latest in genomics and master-assisted selection techniques that are starting to be implemented now in a lot of the universities around the world, particularly at the University of Queensland. So out of all these problems, we decided to take a focus on salinity as one of the first port of calls that we're interested in. And salinity in coastal areas is aggravated by climate change. As you can see on this map here, a lot of low-lying countries are particularly vulnerable, and a lot of islands in the Pacific, and particularly low-lying atolls, are particularly vulnerable to things like cyclonic conditions, tidal inundations and tsunamis and things like that that can affect production. So tidal inundations are a big issue in the Pacific, and particularly affecting places like Coral Atoll Islands. And however, also we have what's called a freshwater lens in a lot of these places too. So this is where a lot of the islands actually have quite large freshwater reserves, even though there are islands surrounded by salt water. But this lens is actually flexible, and when you get cyclonic conditions, you can start to get saltwater infiltration into the freshwater areas, percolating into the root-growing regions of the crops. So salinity could be a real problem in the future if cyclonic conditions are going to continue to increase. So how does salinity actually impact plants? Well, approximately 77% of the world's land area is affected by salinity. 30% of irrigated land is salt affected. You can't take salt out of soil easily once it is there, and it stays there. Salinity dramatically reduces growth and crop yield. So in terms of salt stress, there's seen as two different mechanisms. There's osmotic stress and ionic stress, where sodium accumulation occurs in the plant tissues. In terms of osmotic stress, the speed of onset is quite rapid, but the primary side of visible effect is decreased new shoot growth. In terms of ionic stress, you're looking at a slow speed of onset, and you tend to see increased senescence of older leaves. So you can see here on the picture to my right, an indication of what happens, or what happened in a place in Micronesia on an atoll. You can see here that just all the new growth of the taro has just been devastated. So plant species have a broad range of salinity tolerances. You can see here in this chart, looking at dry matter accumulation as it's introduced to salt, that it ranges from halophytes, which are salt-tolerant species such as saltbush, all the way down to sensitive species such as Arabidopsis and rice. And for taro, based on current research, and I've got to say, as an orphan crop, there's not much literature on taro per se, but in terms of salinity tolerance, there's even less again. So for most of the research we've seen, it suggests that taro is quite salt sensitive, below the 100 millimolar range. But recently there was a paper in 2021 by Lloyd et al. That suggested that taro could survive conditions upwards of 200 millimolars of sodium chloride ion solution, which would place it among the salt-tolerant non-halophytes. So at this stage, that particular paper only looked at one variety of taro, so there's still a lot of work there to be done looking at the different genetic backgrounds and the vast diversity of taro, but it's possible that the taro range could be quite high. But so far, our preliminary research seems it's quite sensitive. So we have done some preliminary studies looking at salinity tolerance with taro. Our very first experiment, we picked a salt concentration from a paper in 1998, which you can see in the below chart here. We decided to pick an upper limit of 40 millimolars, as we thought that would display a quite obvious phenotype. So we developed a 3-factor experiment looking at 2 different taro genotypes, and we had 3 different salt concentrations, 40 millimolar being the upper limit. But we also have become interested, as a lot of researchers are now, using spectral imaging. And of course, a lot of the big players are using sophisticated drones and very expensive thermal imagery. But we thought, why not try to use some of the cheaper stuff that's available? And you'd be surprised what's coming on the market. You may have heard of what's called a Raspberry Pi computer. It's a very basic computer, can sit in the palm of your hand. And these days you can get various versions that even have thermal imaging capacity. It's just a basic camera with a filter over the top. But there's actually publicly available software that allows you to take these sort of thermal images. And we thought maybe we could apply that and see if it can actually be used for cheap phenotyping for things like salinity tolerance or drought. So what was our experimental setup? So we've been using pot systems that have been perfected by Dr. Mel Hunter, as many of you may know. He's developed a lot of great systems that have their own water reservoirs, which in this case, when you're doing a thing like salinity tolerance, it allows you to measure things like water use efficiency, which is very important. And it's a very basic system, as you can see in the picture here. It's just an enclosed pot system that doesn't allow root escape. We grew the plants in sand that had been removed of all salts. Nutrients were supplied. And then we had an upturned 1 litre reservoir that allowed the plant to control the amount of water it used. Because the system's enclosed, it means there's no root escape, so you get very accurate biomass measurements by using mass scales. So you could weigh the plants and get derived root biomass, shoot biomass, and you could also measure how much water usage the plant is using. And you can also see here the system worked very well. Plants grew quite large in the pots, and they also had well-developed root systems. I've also got a picture here of the Raspberry Pi camera. It's very simple. It has Wi-Fi, all the things you'd want in a computer. It has a decent hard drive, and it allows you to take thermal imaging. And on the picture on the right here is an example image of somebody who took a picture of themselves with one of these cameras. And they even have freely available software that allows you to improve the clarity of the image and start taking accurate quantitative measurements. So some of the results we took for some of the phenology measurements we measured, we looked at fresh weight and dry weight. We measured that for the shoots, the roots, also the youngest unfurled leaf and the petiole. Now, not all of those were statistically significant, particularly the root corm and the roots didn't seem to show any distinct differences, but definitely in terms of the shoots and the petioles and the lamina, there was a clear difference when we applied salt over a 1-month period. And as you can see here in the various charts, there was quite a difference between treated and untreated plants. We also measured stomatal conductance and photosynthetic rate using a light core. And as you can see here, it slowly developed until right at the end of the experiment, we got statistically significant differences. But you can see how the range of data starts to change for the salt-treated plants. And what's interesting, as I mentioned before, 40 millimolars of salt gave us a phenotype. But just by looking at the plants visually, it wasn't until after a month that you actually started to see the plants suffer. We can measure changes of water use efficiency very quickly and water usage, but it was a very slow development of wilted phenotypic appearance. So that suggests that maybe that Taro does have higher abilities to deal with salt, and that's what I'll explain in the next experiment that we hope to conduct. In terms of using cheap spectral imaging, this is a picture or a diagram of some of the images that we generated. And we measured various vegetation indices, such as NDVI, Enhanced Vegetation Index, and GCC, which is just essentially a measure of greenness. So a lot of these different indices that these cameras measure give you various measures of greenness. Thermal imagery can detect living versus dying tissue, so they can be quite sensitive. So this is sort of the basic images we had here. Generally, we had originally hoped to have cameras put up on the roof of the glasshouse and capture the whole experiment once, all the plants growing together. But it soon became apparent that in a glasshouse environment, you have various shading effects, different times of the day give different lighting. Then you've got to take into account the fact that taro has very big leaves, and if the pots are close together, they start growing into each other. So even though we could film the experiment all at once, in terms of data analysis, it became quite complex trying to work out which treatment was which. So we ended up doing individual portraits of all the individual replicates. And that was a data that we took on and gave us our final analysis. But eventually it would be good, obviously, in a developing country context, that you could do experiments where you took images all at once. But at this stage, the computer analysis is too complex for us, and we'll still take the data, but hopefully we can extrapolate that future with more expertise. So this is some of the data we generated from the Raspberry cameras. And on the left-hand chart here, this is an image of EVI data. The top chart shows you the salt-treated plants, and the dashed lines mean you've got negative EVI going. And you can see all the salt-treated plants have a lot more negative growth or negative EVI data than the bottom chart, which shows a much wider spread of growth patterns. And you can see here that the plants are a lot more positive with their EVI ratings, which tends to suggest there's a clear difference in growth patterns between salt-treated and non-treated. If you look at the right chart here, looking at the NDVI means, basically, these are all our plants that were salt-treated and control-treated. And the plants that have the pinky brown highlight bars were salt-treated. And you can see we applied salt in the very first week where the asterisk is. And as you go through, you can see that the NDVI means are starting to move to the left for all the salt-treated plants, but the non-treated plants are growing just fine. So this was exciting results for us. It showed that, in fact, these cameras could detect something, and it seems to suggest that they can detect some sort of phenotypic difference. But obviously, this is very early days, and we need to do much more work to be definitive about that. So this leads me to talk about the main project that we're focused on. And it's a small project that we managed to gain funding from the Australian and Pacific Science Foundation. And this project is going to be running from 2021 to 2024. And essentially, we asked the obvious question, is there genetic diversity for salinity tolerance that could be utilised for crop improvement? Obviously, soil salinity is already impacting island nations such as Tuvalu, Kiribati, coastal areas of Fiji and Samoa. And because of quarantine issues, we can't really do direct measurements on taros in the Pacific because they've got their own projects doing this work. But we developed our own idea that perhaps we could look at wild relatives of taro growing in Queensland, particularly in South-East Queensland waterways, such as, for instance, there's native taros, such as Conjuvoi. If anyone's been to Cunningham Gap, where they've got the marker for where the explorers first went through, there's a lot of native taro growing around that statue. But anyone who's gone around, walking around the waterways of South-East Queensland, you actually will see taro everywhere. So you can see in this bottom picture, there's a lot of Colocasia esculenta, which is the edible form of taro. But in fact, this naturalised taro is considered a declared weed in most of the waterways around Australia. So it's been growing for many years now, and we thought this could possibly be a source of salt tolerance, the idea being that taro growing in the upper reaches of the river system would be growing in more freshwater environments. But as you get further down the streams towards the bay or the marshlands or things like that, or wetlands, you're going to get more brackish salt water. And it'd be interesting to see if taro could still grow in these environments and whether over time these taros have adapted and actually developed some sort of mechanism of salt tolerance. So that's the basic premise of this project. So what are the main scientific objectives? Well, firstly, we'd like to phenotype the Australian taro wild relatives for salinity tolerance, particularly focused on the weedy naturalised taros growing prolifically throughout the waterways. We'd like to identify key sources of salinity tolerance. And then ideally, we'd like to undertake DNA fingerprinting and GWAS analysis to see if we can find any genes or QTLs involved in those mechanisms. And if we're completely successful with this project, the big plan is to juice some of this germplasm back into the Fijian collection, and hopefully they can make positive use of that in the future if they ever need it. And like any projects that are working with developing countries, there's an aspect of capacity building that we're involved in. And we'd like to strengthen the relationships between UQ, SBC, and the Pacific partners. And to this end, already, Gemma, our PhD student, has won a Crawford Fund. And as you can see in the picture in the top here, she's travelled over there and she's learning all about the industry, how to characterise taro, its phenotypic descriptors, how it's grown, how the industry runs there. And she's able to not only impart some of the knowledge she's learned here in their labs and how to do conduct genotypic fingerprinting, but also they've been teaching her a lot about how the industry's run and how we can sort of formulate our own experiments to make sure they're in keeping with best practice for farmers. So obviously the first tranche in this project is to collect wild taro populations. And we're aiming to collect a population of around 250 plus. The numbers are chosen particularly to be statistically relevant for a GWAS analysis. Obviously, we have budget limitations, so we're trying to maximise that as much as we can. But that's a general idea about the numbers there. And one of the great technologies we found, and have been developed a lot lately, is there's a lot of websites and publicly available apps such as iNaturalist, which we're making use of. And this software or basically website allows anyone with a mobile phone, if they download the app, to go around and take pictures of any plants or animal specimens they see. They can botanically name them. And then the good thing about it is it puts GPS coordinates about wherever you detect something and puts it all into a database or a map that anyone can access. So we can effectively, a lot of you or some of you may have seen that we were in Gardening Australia not too long ago, canoeing around the riverways and collecting taro. And basically, this app allows us to catalogue all the taros we find. We can GPS locate it. And then anyone in the public can also take pictures of taro wells. So if you want to download the app, I hope you do that. And you can help some science projects go forward, such as our own. So besides just collecting the taro, we also want to characterise the population's morphology. We'd like to collect leaf tissue for virus indexing, which is important to make sure that it's clean material if it's going to be given to the Fijian SBC collection. But we're also obviously going to be taking pore water samples to check the EC content or the electrical conductivity or salt content of the samples we take all around the waterways, with the hypothesis being in the upper ways, it's going to be more fresh water, but as we get further down, it's going to be more salt involved, or more salt affected, I should say. So how are we going to screen these plants for salt tolerance? So when you're looking at a population of 250, you also have to account for biological and technical replicates. So that ends up being quite a lot of plants that need screening. So we needed a high throughput system. And taro generally is grown in almost like rice in sort of a paddy system. They call them pits, but it's in irrigated, fully irrigated growth patterns. They can also be grown in rain-fed, but we knew that in the hydroponic system, taro would probably do very well. So in Gatton here, we've developed what's called a bucket system. It's very simple. In pots of sand, the taros potted, it sits in the bucket, and you've got constant irrigation of nutrients and water, and that's recycled and goes through. And you can see in the picture in the top left here, we've got 2 reservoirs, and one reservoir is going to apply plants that are our control plants, and then the other reservoir is going to be our salt-treated reservoir, and we've decided to pick a salt concentration of upwards of 100 millimolar, which is sort of the midpoint from that latest paper that says can go all the way up to 200 millimolar. We've already shown at 40 millimolar, you can detect a phenotype of salt tolerance. But we're hoping at 100 millimolar, we can speed up our experimental process and get things done a lot faster. So what sort of detailed phenotyping do we intend to do? Well, obviously, we're going to measure things like phenology, plant height, leaf number, leaf area. We'd also like to measure somal conductance, and we're also going to measure above and below ground biomass. You can see in the bottom right-hand picture here, in Fiji and Pacific Islands, whenever they do experiments, they use what's called a headset, which is basically they cut off the top of the corm and they just leave one shoot. The reason they do this is obviously when you're doing experiments, if you're doing things with cereal crops, you can grow things from seed, so it's easier to get uniform plant size. But when you're doing a vegetatively propagated crop, you need to think a lot harder about how you can get uniformity in your plant size before you start experiments. And this is one way we hope to do it. Also because that's the industry standard that's used overseas. We also obviously want to also investigate the low-cost phenocams again. We're going to be measuring various indices of greenness plus thermal imaging. And I just want to point out what's great about these cheap cameras, they also come with a lot of people that post their data analytical techniques to a thing called Public Lab, which is found in California. And a lot of this publicly available resourcing means that countries such as the Pacific Islands and Fiji can hopefully start to do, if we can validate the system, a lot more complicated phenotyping, but there's also a lot less expensive to do, where you have to do all this detailed phenotyping. You might be able to get away with a lot of low-cost phenocams. In terms of the DNA fingerprinting and the GWAS, our hypothesis is that a lot of the material, especially the weedy material in the waterways, is going to have low diversity. In fact, a lot of it could be clonal because a lot of the plants can just bud off and travel downstream and regrow again. But until we do the microanalysis, we won't know that. But we do expect, hopefully, to find some differences between plants growing in different environments. Concomitant to the DNA fingerprinting work we had to do, we're going to be using, we're going to use dart seek markers, which is quite well utilised at UQ. And there's a big body of knowledge there behind using that technology. But we also hope to measure flow cytometry, looking at the chromosome number for a lot of these plants, just because it's not known. But there's also a population genetics angle, looking at this work, looking at how a weedy plant grows throughout South-East Queensland waterways, how it's evolved. So looking at the chromosome numbers there to see if it's all diploid or if there's actually triploids would be quite interesting. So what's the progress to date? Obviously, the floods affected us quite a lot. The glass houses were destroyed, as everybody knows. Taro is one of the few plants that actually thrived during the floods. So we didn't actually lose our planting material. But what we didn't realise at first was that when you look at this picture to the left here, a lot of the actual waterways were completely scoured of vegetation from the flooding. It was so powerful, it removed all the vegetation from the creek banks, which means a lot of that taro that we hoped to collect was washed away. That doesn't completely ruin our experimental plan. It just means we're probably going to have to work a lot harder to find taros for our collection. In saying that, though, I just wanted to show you that if you look in the far right picture here, we have a taro growing amongst mangroves, which is quite exciting. And in fact, we found taros growing in the Minipi wetlands, Stones Corner, a few places that shows the taro can, in fact, grow in salt-affected environments. Obviously, we found Taro everywhere in the upper reaches of the Brisbane River, and it gets a lot less prolific the further you go down, but you can still see Taro growing in brackish conditions. In terms of our hydroponic system, and you can see in this picture here, we've now got it pretty much optimized. Originally, we found out that because this greenhouse was not ventilated, that temperatures were getting up to 40 degrees and the Taro was not doing very well. But we've since been able to improve that. We've put insulation on the sand surface to reduce evaporation. We've got a lot more flow in our hydroponic system, and the plants are growing a lot more happily now. So we're quite confident we can start to begin experiments. And hopefully in the not too distant future, Jamri, our PhD student, will have all the data and she'll be able to present and start answering some of these important questions that I've raised today. But thanks for listening to my presentation. I'd just like to thank our funders, the Australian Pacific Science Foundation, and I'd just like to acknowledge quite a few people that have helped make this project possible, particularly Millicent Smith, who helped me gain the funding in the first place. Has been a great source of knowledge in terms of the physiological parts of our experiments. Gemma, our PhD student, is doing a fantastic job. She's already travelled to Fiji and has learned a lot of knowledge there, which has been invaluable for our work here and has encouraged collaboration with people in Fiji. Ian, who's always had a background in genetics in the Pacific, has been invaluable, creating connections and even gaining us access to funding and things like that. I'd like to thank Mel Hunter for some of the pot systems he's been able to educate us about. I highly recommend people who are forced to work in glasshouse conditions. There's a lot of optimization that can be made in the sort of pot systems you use, and some of them can be quite successful. They're not as good as the feel, but with a lot of thought, you can actually do a lot better than just the basic plant in the pot with watering. I'd also like to thank Dr. Beth Addison-Smith from QT, who I originally met doing a lot of allergy work with phenocams, looking at grass pollen shed. She's been invaluable. She's a computer scientist and has a lot of database knowledge and data management knowledge. I'd also like to thank Scott Chapman and his team, particularly his postdoctoral team, who we hope to utilise in the future with some of the more sophisticated data analysis, looking at some of our phenocam data. But thanks again for listening to my talk, and I'm happy to field any questions if there are any.
Yeah, thanks for that great talk, Brad. Really interesting stuff. We've got a question here from Jane O'Sullivan. She's wondering, what is the state of Taro breeding these days? And if you could let us know if there's any success stories from introducing traits from wild or non-preferred cultivars. She states that, getting them to flower and to seed has always been a huge problem, which makes breeding difficult.
So Jane is entirely right about breeding Taro being difficult. From my understanding, Taro seed is only viable for 28 days. So there's a very short window there for doing crosses or even for maintaining seed. That's why it tends to be vegetatively propagated. Because of the long maturity times, there's also vegetatively propagated. But in the past, there has been breeding programs. But in a lot of developing country contexts, you find that the biggest issue is just maintaining staff or people with expertise. In the Fiji and Pacific Islands, previously they've had breeders there, but the breeding programs tend to stop and the breeders tend to move on. So continuity has been the biggest problem. I believe the University of Hawaii probably still has a breeding program, but I know in the Pacific, there's nothing really going on at the moment. So it tends to be on and off in terms of breeding. So part of our future plans are, once the entire collection of the in vitro germplasm collection in Fiji is being genotyped, we hope to develop some sort of phenotype prediction models and genomic selection ideas, particularly to sort of compensate if a dedicated breeder can't be there to make improved germplasm for some of these problematic traits like drought or salinity tolerance.
Mm-hmm. Thanks for that. So in the plants that are grown, you said that they can be diploid or triploid. Do you know if some of the preferred varieties are triploid, or is it mostly diploid plants?
I think the vast majority are diploid, but I think there's quite a lot of interest in triploid plants per se, just because of the fact that they are sterile and they would be quite important to their own collection and that sort of thing like that. There have been studies done on the ploidy of Taro, but not since the late 1990s. So this is an area that would be interesting. And it's also interesting to see what sort of ploidy would look like in a weedy plant context in South-East Queensland waterways, I suppose.
Yeah, absolutely. It'd be interesting to see what we sort of have around the country once you guys get started. Maybe just to end it off, what do you think is the greatest limitation to progressing this sort of work right now, and how do you plan to remedy that?
Well, I mean, I guess currently, so in the Pacific, they've maintained this germplasm collection for quite a long time, but there is a vast dearth of information in terms of phenotypic information about their collection. So just basic phenotyping to characterize what they have is important. Just understanding the growth habits, the different phenotypic descriptors, and maybe now they're in the good position that a lot of money is flowing from Australia and New Zealand to do a lot more work in the Pacific, that they can actually harness this to sort of build their database up. And I guess the grand plan would be that they would have a world standard database of Taro that is genotyped, and then they can start to infer the properties of these plants. And then they can start, I mean, one of their main job descriptors is to give lead plant material to farmers. So if they have a better idea of what they have in their collection, they can start to go, well, we believe these particular varieties have these qualities, such as salt tolerance, and they'd be good in this context. And you can start to really provide a benefit to the industry. Perhaps in future, they might have a dedicated breeder and you can start to actually really make some genetic gains. But at this point, just sort of cataloging what they have would be a huge step forward.
Yes, most definitely. Okay. Well, thank you for giving us such a great seminar today, Brad. For everyone who's listening, if you haven't already signed up to the seminar invitation list, please do that. Brad just put it up there, or email these people. For next week, we've got someone from the University of Nebraska. We've got Dr. Melanie Hess, who's going to be giving a talk on improving the microbiomes for prediction of methane in New Zealand sheep, which should be great. So hopefully they'll come back again next week. So thanks, everyone.